Good morning. This is the first video for your series of videos for April break. Something to keep in mind is that after this April break, when we return, we have approximately 6 classes together before the AP test, and those will pretty much solely be review. And so all of the kind of learning that you do over break is on your own, and we will take a moment to review all that when we get back, but then we'll dive right into review for the AP test. This first section of videos is about gene expression. If you remember, there was an open response on gene expression on the AP test. It was in a plasmid or a circular DNA, and it's one that we struggled with a lot. It's actually a concept that I struggle with a lot as well, and so we're going to talk about prokaryotic gene expression, eukaryotic gene expression, and then talk a little bit more about how that happens and how we control it. So when we're talking about the prokaryotic regulation of genes, this is an example. It is the tryptophan synthesis pathway. And so we have to keep in mind that for every gene, right, every section of DNA, it codes for a specific protein, either an enzyme or a protein that does something in the body. Tryptophan, right, is a chemical or a protein that's produced in the body when we eat certain foods. Similarly, when we eat turkey on Thanksgiving, we talk about this tryptophan, or the chemical that makes us really sleepy and tired. And so this segment of the DNA, this TRP gene, codes for that specific protein. So this produces something that will interfere with the function of the enzyme in a pathway. It produces a gene regulator that can inhibit the transcription of one biochemical pathway enzyme. And so there are kind of two ways that we get negative feedback, or we get the stop of this pathway. When we talk about regulation, what we are looking at is, how do I slow down or stop the production of an enzyme? Because otherwise it will continue making that protein. And again, the two ways are that it'll either produce something to interfere with the function of the enzyme, so once that enzyme tryptophan is made, it will stop it, or it will produce a gene regulator that stops the transcription altogether. Eukaryotic cells have many more genes, and their genomes are quite a bit larger, so therefore the regulation is a little bit more complicated. Physically, there are more obstacles to regulate eukaryotic genes because there's so much more DNA to manage. In addition, there are other non-histone proteins that are used in eukaryotic gene expression. And so when we talk about the regulation of genes in a prokaryotic cell, it's a pretty simple process. So when we look at a gene or a section of DNA, we have our operon. It's a group of prokaryotic genes with a related function that are often grouped and transcribed together. The operon has one promoter region, if we remember the TATA box, and it transcribes multiple genes. So if we look, we see the TRP, E, D, C. Those are all specific proteins, and they are often coded together so that they can all work in tandem. An operon, or a section of genes that are related, is made up of the following things. The actual structural genes. These are the genes that are related to use in the pathway, so TRP, E, D, C, B, A. The promoter, which is the nucleotide sequence that binds to RNA polymerase to start transcription. We saw it as the TATA box in our practice with transcription and translation. And then the operator, which is the nucleotide sequence that can bind with repressor proteins to inhibit transcription. So if we think back to the first slide where we talked about there's two ways that we regulate, or two ways that we can stop transcription. The operator, which is the segment right after the promoter or the TATA box, can stop the transcription of these genes. The regulator gene, this gene produces a protein called a repressor that can inhibit the transcription of an operon by attaching to the operator. So this is the order. If we look at that DNA sequence, regulator gene. If that gene is transcribed, it's going to make a protein that will bind to the operator. Then we have our promoter, which is our kind of ta-ta box, and then we have our operator. Repressors have something called allosteric properties, and this is an important concept. These modulators, or these little proteins, can bind to a repressor at an allosteric site, changing the conformation of the repressor, therefore activating or deactivating the repressor. So if we look, the repressor is the little red protein, and that's what's made by that gene, that regulator gene. It will bind with the operator. If the protein, that little inducer, is present, it's going to bind to the base of my regulator protein, and it's going to bend it essentially so that it will not fit into the operator. This will keep the genes, it will promote the genes being made more frequently. If I have my regulator protein and it's bound to another type of protein called a co-repressor, it's going to straighten that protein out. It's going to allow it to bind to the operator, and it's going to inhibit the production of those genes. So the lac operon is an example of an inducible operon, and this shows the concept that we just talked about. So we see that LAC-L is my regulator protein. It makes my little red protein shown below. If allolactase is present, it will bind with my red protein. It bends and it makes it inactive, so it doesn't bond with the yellow operator. This means that my LAC-Z, LAC-Y, and LAC-A proteins are continually being made. If no lactose or allolactose is present, the receptor or the repressor protein is active and it binds to the operator site. This prohibits the RNA polymerase from transcribing. So I want us to think about this in very real terms. If I eat dairy, right, I'm sitting here, it's 6 in the morning, I'm drinking my coffee and it has cream in it, and so therefore I'm ingesting lactose. This situation is occurring. That allolactose, that green thing I'm ingesting, it's binding to the protein that's being made, and it's prohibiting it from bonding with the operator, the yellow segment, and so my LAC, Z, Y, and A proteins are being made, and they're going to work digesting the lactose that I've consumed. Eventually, once that's processed, I'm not eating any more dairy. There is no lactose present, therefore the repressor is going to bind to the operator, and those enzymes are not being made anymore. So these are animations. If you want to see these in class, please let me know. I'm happy to show them. So this is kind of the synthesis of tryptophan and the repressible TRP operon. So this one works a little bit differently, and so what we have to keep in mind is that each segment of DNA and how it is regulated functions differently. In this case, we see that tryptophan is absent and the repressor is inactive, and so the body is going to continually make TRPE, D, C, B, and A proteins. Tryptophan is absent. If tryptophan is present, it's going to bind to the protein, my repressor. The repressor is going to bind to the operon, and I'm not going to make any more. So this is the reason why. I naturally make a certain amount of tryptophan in my body all the time. If that is happening and I'm not getting any extra sources, my body will continue to make it. The minute that I consume food with more tryptophan in it, this situation is going to happen where it's going to stop the production of my natural tryptophan. It's going to use the synthetic or the ingested tryptophan. The LAC and TYP operons are examples of negative gene regulation, as the repressor protein inhibits the transcription, right? So when we saw that red protein bind, it's going to inhibit the transcription of the operons. That is called negative gene regulation. There is something called positive gene regulation, and as you can imagine, it means that the repressor needs to bond to have this occur. So this scenario is when I have lactose present, but I don't have any glucose. What's going to happen is I have cyclic AMP instead of ATP, and it's going to promote the transcription and translation of these genes. When I have lactose present and glucose present, cyclic AMP is low, and there's very little lac synthesized. So the next set of videos that we're going to talk about is eukaryotic regulation, and I'm actually going to pull that up now so that we can kind of consolidate videos for the sake of your sanity over this break. So we've talked about that there are just two really simple ways to regulate prokaryotic DNA, right? We either get in the way of the enzyme working or we prohibit the DNA from being transcribed. It's really simple, and we looked at positive and negative impacts on transcription and translation. Now we're going to talk about eukaryotic gene expression, which is far more complicated because the DNA is a lot larger, and by larger I mean there's much more of it. There are many more genes, and the controls are a little bit more complicated. Again, you've seen this slide before. This is why they are so much different. Eukaryotic gene regulation. Almost all cells in the organism are identical or totipotent, right? Identical cells and everything. Differences between cells result from differential gene expression, the expression of different genes by cells with the same genome. So all of your cells have identical DNA, but which genes are turned on determines if it is going to be a heart cell or a skin cell or anything else. Errors in this gene expression can lead to diseases, including cancer, and gene expression is regulated in many stages. So again, just as a review, our organization of DNA, right? We have our strand of DNA at the very top. We have the DNA wrapping around nucleosomes, or wrapping around histones, which are the little purple beads, and they make nucleosomes, which are those kind of big clusters. Those wrap up into our chromatin fiber. Those start to loop into protein scaffolds. We have them start to condense and become chromosomes that we usually only see during metaphase. Normally DNA is not wrapped that tight. Types of repetitive DNA. So there's a lot of non-functional DNA, or a lot of DNA that we don't know the cause of. So DNA is very repetitive, and so this data kind of looks at the amount of DNA that is tandem, so long lengths of repetitive DNA, and then interspersed repetitive DNA, so similar stuff but repetitive. So between the tandem and the interspersed repetitive DNA, that makes up about 50% of our DNA, right? We see that there's about 40% interspersed and about 15% all repetitive right in a row, and so that means that that's a lot of stuff that is not gene DNA, and we'll talk about why a little bit later. We talk a lot about how we alter genes, and it's technically a form of gene regulation. We're going to pause here and we'll pick up in the next video.